Next, we're going to look at the chemistry of visual pigments. We have a couple of players when we're looking at the chemistry of visual pigments, one being retinal. Retinal is just a light-absorbing molecule. It can combine with one of 4 proteins. We're going to look at rhodopsin, which is one of those proteins. All of the proteins are called opsins, and we're going to really basically combine this light-absorbing molecule with one of the opsins to form visual pigments. These visual pigments are synthesized from vitamin A, and they have a couple of different isomers or different forms. There is the 11-cis-retinal or bent form, and the all-trans-retinal or straight form. And bent and straight is just talking about their molecular orientation. The bent to straight form change is going to occur when the pigment, so rhodopsin, absorbs light. And we're going to see that this conversion of bent to straight is going to initiate reactions. The reactions are going to be ion channels opening, but that will cause an electrical impulse because we'll get a depolarization or an influx of sodium through those ion channels. So the overview that we're going to be talking about is a series of steps. These are molecular steps. And you may think that we'd have to turn a rod or a cone on in order for the brain to process that visual information has been received. But actually, if we're looking at a rod, you turn the rod off in order to say that visual information has been received, and that allows the rod to inform other cells that light is present. And that's all the brain needs to comprehend visual stimuli and pick up that information from the photoreceptors as it travels to the ganglion cells, then to the central nervous system. So that's our overview. We're going to see again that turning off the rod actually turns on or notifies other cells of the presence of light. And that's going to allow the brain to realize there is light and comprehend what is being viewed in order to make sense of the world. So again, retinal changing shape from bent to straight starts a transduction cascade of events, and that allows the signal not only to travel via the optic nerve, but reach the brain and be processed by the appropriate cortices. And that whole process is phototransduction, or the phototransduction cascade. And that's just a process where we turn light energy into chemical energy, that kind of chemical electrical energy being a gradient potential then an action potential. And the key is that light-absorbing molecule, that retinal, that sits within an opsin. We're talking about rod, so it's rhodopsin. And retinal is just in a part of rhodopsin. There's many different discs inside each rod, and they're able to pick up on, retinal is, on the wavelengths of visible light in a certain spectrum. So really kind of shades of gray, kind of the tones you associate with peripheral vision. Of course, cones also exist. There's blue, red, and green cones, and they're named for the color of light that each cone absorbs best, so each cone variety. So color perception is possible by activating more than one cone and kind of mixing the colors together. But again, our main focus here, as an example, for photoreceptors and how they work, and therefore phototransduction, is rods. And rods contain retinal. The opsin they contain is rhodopsin. So we're looking at the light-absorbing molecule retinal sitting in rhodopsin. And there's plenty of these discs that contain rhodopsin and retinal in a rod. So that's what we're looking at here. Now this is just the tip of a rod. We can see these mini discs. They're called rod discs. And we look at just one of the rod discs, and just at its upper cell membrane, we have a lipid bilayer that contains protein components, or an integral protein, and this integral protein is rhodopsin, and in rhodopsin is this chemical retinal. So retinal is our light-absorbing molecule. There are 3 steps to phototransduction, or capturing light: pigment synthesis, pigment bleaching, and pigment regeneration. So first and foremost, we have to kind of accumulate rhodopsin, and it accumulates in the dark. Then, when rhodopsin absorbs light, so retinal and rhodopsin, it causes retinal to change shape, so it goes from bent to straight. And if retinal changes shape, then rhodopsin changes shape, and that causes them to separate, so they're no longer going to associate with each other during pigment bleaching. So that causes rhodopsin breakdown. And then we get pigment regeneration. So after basically we've exposed the rod to light, thereby turning it off, but signaling the brain that light has been experienced or captured by retinal, we have to kind of reset. We want to regenerate the pigment, and then we'll go back into pigment synthesis where it accumulates. But we have to first turn the all trans or straight retinal back into the bent form or 11-cis isomer. And those rhodopsins are going to be regenerated in outer segments, and then they'll kind of populate the rest of the segments. And that's what we're seeing here is those 3 steps: pigment synthesis, pigment bleaching, and pigment regeneration. So again, we're going to turn off the rod when it's exposed to light. Retinal, which is in rhodopsin, is going to go from bent to straight, or 11-cis retinal to all trans retinal. And then we just have to reset. We have to regenerate that pigment, and then it'll kind of accumulate in the dark again during pigment synthesis. So essentially, what we're seeing is light energy is providing enough power to cause retinal to change shape from bent to straight, and then retinal changing shape causes rhodopsin to change shape, and that begins a series of events. So that's also going to include a second messenger system that opens cation channels and allows the influx of sodium ions. And that influx of sodium ions is going to cause a depolarization in the retina and the photoreceptors. And that's going to create a gradient potential that, if strong enough, can go to the first order neuron and then travel the rest of the processing circuit. Next, we're going to move on to eye infections. We'll just look at them briefly. Just be aware of them, understand their definitions. So a chalazion is an infection of the tarsal gland, which is under your palpebrae, and that's going to result in a cyst generally. A stye is inflammation of any of the other smaller glands, so not the tarsal gland specifically. Watery eyes can develop because of nasal inflammation that constricts or blocks the ducts, like the lacrimal or nasolacrimal duct, and it prevents your tears from draining. And then conjunctivitis is inflammation of your conjunctiva, which is that anterior covering of your sclera and the pupil, and then also lines the inside of your eyelids, but that's also known as pink eye. It can be bacterial, it can be because of allergies, it can even be viral. Some disorders of vision include diplopia, strabismus, retinal detachment, glaucoma, cataracts, color blindness, and night blindness. Now, double vision is also called diplopia, and that's when muscle movements are not coordinated, those extrinsic muscles of the eye, for some reason not able to coordinate like they're supposed to. Strabismus is another name for someone being cross-eyed. That's generally weakness of eye muscles. It occurs usually in one of the eyes, but it can occur in both. Retinal detachment can occur if the pigmented and nervous areas of the retina separate. This could occur if aqueous humor builds up between the neural and pigmented areas. Glaucoma is when pressure, maybe from aqueous humor, puts pressure on the optic nerve and can result in blindness. We've talked about a cataract before. It's usually clouding of the lenses. It can be surgically corrected, but usually comes back. It's sometimes a complication of diabetes mellitus or just old age. Color blindness is when there's a congenital lack of one or more cone types. So someone's missing a cone type, so they don't process all levels of color well. Red to green color blindness is very common, or the most common. Night blindness is when something's wrong with rods, so rod function is hampered, and this could be because of a vitamin A deficiency. So vitamin A is something that rods use in order to help create the chemicals it needs for phototransduction. Next, we're going to move on and look at our chemical senses. So taste is also called gustation, and smell is also called olfaction. Both taste and smell use chemoreceptors, and these chemoreceptors only respond to chemicals if the chemicals are first dissolved in an aqueous solution. So think about that. For taste, we have chemicals dissolved in saliva. For smell, might be a little hard to think about, but we actually dissolve chemicals that we smell or that get into our nasal cavity in a mucus layer that covers the olfactory epithelium at the roof or the top of our nasal cavity. If you wanted to do an interesting experiment, you could take a paper towel and dry off your tongue and put a little bit of sugar on your tongue. And you'll notice that the dry tongue is not actually able to process the sweet taste until saliva returns to your tongue. Very simple, quick experiment to test the fact that we need an aqueous solution to dissolve chemicals in order to taste or smell. Now we'll start with smell. So olfaction. The organ of smell is your olfactory epithelium. This is a series of cells that is found at the roof or the top of your nasal cavity. They contain, again, a number of different types of cells that are generally sensory neurons. There's olfactory receptor cells, supporting cells, and basal cells. So supporting cells do exactly that. They surround, they cushion the olfactory receptor cells. Basal cells would be closer to kind of the basement of this membrane, which is further away from the roof of the mouth that you'd be able to see. So these basal cells lie at the base of this olfactory epithelium, and they're kind of like stem cells. They'll help regenerate the receptor cells if needed. We're most interested in the olfactory receptor cells, which are bipolar neurons, and they have radiating olfactory cilia. And it's these mucus-covered radiating olfactory cilia that pick up on the chemicals dissolved in fluid. Then there's bundles of axons of olfactory receptor cells that form the olfactory nerve or the cranial nerve, and we'll look at them and how they synapse with the olfactory receptor cells. So here's a general overview of what we see for the olfactory epithelium. So we have the olfactory bulb that is at the base of the brain, and it is sending out neurons through cribriform foramina, which are just tiny holes. So if you're familiar with the cribriform plate, when you went over the skeleton and learned parts of the skull, the cribriform plate is where the olfactory bulb is sitting, and the cribriform or olfactory foramina, those are the holes that the olfactory nerves go through to synapse with the receptor cells. But we can see the olfactory epithelium is located at the roof of the nasal cavity. So here's a better view. We've kind of just blown up that olfactory epithelium. So we can see our mucus layer. This would be kind of the open area of our nasal cavity. So all this is up top of the nasal cavity. You can actually see the bone up at the top of the nasal cavity here and the little holes. So those are our foramina. But let's go down to our mucus layer again. We have our cilia of our olfactory receptor cells sitting in that mucus. They're bipolar cells, so this is their receptive end. Then we go to their cell body, and then they have an end or a central process that goes towards the central nervous system. It's that end that's going to synapse with a mitral cell. So a mitral cell is going to be located in the olfactory bulb, and where they synapse, this is called a glomerulus because it's just a very broad synapse. It has a lot of kind of dendritic endings and then axon terminals, and it kind of looks like a ball of yarn. That's where we're getting the term glomerulus. You'll see glomerulus again when you get to A&P2 and talk about the kidneys. So we have our olfactory receptor cells synapsing with mitral cells in the olfactory bulb. The essentially axons of the olfactory receptor cells are going through the foramina, called olfactory foramina or sometimes cribriform foramina, and they're forming essentially filaments of the olfactory nerve. We have an olfactory gland helping us secrete this mucus layer. We have our supporting cells in between our olfactory receptor cells. And then in the basement layer of this epithelium, which is kind of reversed if you're thinking of it, because here's the basement membrane, here's the apical layer that's near the open or free space. These basal cells, they could replace the olfactory receptor cells if they were damaged, which you can damage olfactory receptor cells. I had plenty of chemistry teachers that had accidentally inhaled one too many chemicals that were caustic or just spent too much time in the lab before we had all of the necessary kind of safety precautions, and they didn't really have a great sense of smell anymore. So most of the time when you damage them, give it 24, 48 hours, they'll regenerate. But you can damage them beyond repair. The physiology of smell is pretty simple. So olfactory receptor cells respond to different chemicals in different ways, but the olfactory receptor cells synapse with mitral cells of the olfactory bulb. So after the signal's processed and it causes the opening of ion channels via second messenger system, and sodium influx occurs and a depolarization is made, the depolarization of the olfactory receptor cell membrane is going to trigger an action potential in the axon of the receptor, which is then communicated to the mitral cell in the olfactory bulb. So it's really the mitral cells that process the odor signals and then help send that impulse to the brain, first to the olfactory cortex, but also to higher levels such as hypothalamus, amygdala, and limbic system. And that's again why we link emotion and smell and memory together, is because of the ability to send this signal to not just the proper somatosensory cortex, but also other areas of the brain. Next, we're going to look at gustatory sensations. So taste is next. Taste is also due to a chemoreceptor. We're going to see that a chemical is dissolved in saliva. The sensory cells for taste are taste buds. Well, specialized cells in taste buds, I should say. Taste buds themselves are really just kind of bumps on your tongue. There can be some taste buds on your soft palate, inner surfaces of the cheeks, your pharynx, which is your throat, and your epiglottis, but most of them are going to be on your tongue. And 80% of taste perception is actually smell, so smell and texture definitely enhance or detract from taste. But if we try to get down to the base taste sensation, you do need the sensory receptor cells in taste buds to process the chemicals that we consider taste. But we also have to include thermoreceptors, mechanoreceptors, nociceptors, because even though it's hard to explain, if you burn your tongue, you can if you've done it before, you can immediately think of kind of what it tasted like or the sensation that occurred when you burned your tongue. So even though that's a pain response, it does influence taste. If the pain response is strong because you really burned your tongue, it actually can dull taste for a period of time. So let's just look at the taste buds quickly. You have a couple of different types of taste buds. Again, taste buds are bumps on your tongue. We have fungiform papillae, circumvallate. There's also other ones called filiform, but filiform are the only bumps on your tongue that don't contain taste buds. All the other taste buds do contain taste cells. But again, each papillae contains many taste buds, but your filiform papillae, those bumps, not really, no sensory cells. But just like our smell, we have supporting cells. We have basal cells, and then we have our receptor cells. In this case, our receptor cells are gustatory cells. So gustatory cells are our receptor taste cells. Basal cells are stem cells that could replace damaged gustatory cells, and supporting cells help insulate the receptor. We can see over here on the right many types of bumps on the tongue, mainly anterior to the circumvallate. Again, papillae are bumps on the tongue. Papillae contain taste buds unless it's the filiform papillae. They do not contain taste buds. Then if we blow up one circumvallate papilla, we can see that there are crypts on the side. And in those crypts of everything but the filiform papillae, you would find taste buds. So we're going to look at one of these taste buds on the side of this tongue bump. So let's go over here. Here is one taste bud. There's a taste pore, so that's how the chemical could get to the taste cells, the gustatory cells. There are gustatory hairs or cilia on the gustatory cells. And when that is bent or moved or processes chemicals, it opens ion channels that creates a depolarization signal, which can help generate an action potential if that receptor potential is strong enough. So you can actually see that these receptor or gustatory cells are communicating with another neuron. We have our supporting cells in between the gustatory hairs, or cells of the gustatory. And then we also have our basal cells, our stem cells. So similar in structure, if not in function. There are 5 taste sensations: sweet, sour, salty, bitter, and umami. And it just depends on what type of chemicals are being dissolved in saliva and then processed by gustatory cells. So sweet includes different types of sugars or saccharins, alcohols, some amino acids. They're going to be processed as a sweet flavor. Hydrogen ions are processed as sour. Metal ions taste salty. Alkaloids such as quinine and nicotine are bitter. And then umami is amino acids, glutamate, and aspartate. If you're not sure what umami is, that's kind of the MSG or kind of meat taste in a lot of foods. The physiology of taste, also pretty simple. First and foremost, as a chemoreceptor using sensation, we have to have the chemical being tasted dissolved in a solution. For taste, that's saliva. That chemical dissolved in a solution must reach a taste pore and come into contact with the gustatory hairs of the gustatory cells. And then once we have the binding of the food or the chemical, we're calling here a tastant. In smell, it was called an odorant. Once that tastant binds to the gustatory hairs, we're seeing that depolarization of the taste cell membrane, we're going to cause release of a neurotransmitter, communication with another neuron at a synapse, and that will at first generate a receptor potential, and that receptor potential can become a generator potential in the next neuron, and then elicit an action potential. The gustatory pathway is towards the brain. So we're going to use cranial nerve 7, or the facial nerve, and the glossopharyngeal nerve, or cranial nerve 9, to carry impulses from taste buds to the medulla oblongata of the brainstem. Then from the medulla oblongata of the brainstem, the impulse is going to travel upwards towards the thalamus, which is the relay station for the brain. And then from there, the fibers are going to branch to the appropriate somatosensory cortices, including the gustatory cortex and the insula, or the insular lobe. We're also going to send signals to the hypothalamus and the limbic system. So just like with smell, we send gustatory signals to other parts of the brain as well. And this allows for the appreciation of taste. The vagus nerve can transmit from the epiglottis and lower pharynx if it happens to be a taste bud located there that is sending the signals to or up to the thalamus to be relayed to the gustatory cortex. The gustatory cortex, again, is going to be the main place that we're going to kind of process taste, but it communicates with the hypothalamus and the limbic system. There's also reflexes controlled, for example, by the hypothalamus, like the secretion of saliva, gastric juice secretion, gagging, vomiting. That has to do sometimes with taste, but here you see the basic gustatory pathway, so from the tongue to the medulla oblongata, up to the thalamus, and then out to the insular lobe, where there is the gustatory cortex. Next, we're going to talk about some disorders of chemical senses. There's agasia, uncinate fits, and anosmias. So agasia is loss of taste, anosmia is loss of smell, and then uncinate fits are olfactory hallucinations, usually very unpleasant, something like the smell of rotting meat, or kind of a light hallucination that causes, not necessarily a light hallucination, but similar is light hallucinations that cause headaches. But the most common reason someone may have an uncinate fit, which is a smell hallucination, like the smell of rotting meat, is because they're about to have a seizure. So it is a common symptom that some individuals have, is some type of hallucination or some type of feeling that they're about to have a seizure if they suffer from those. And this would be kind of an aura or a clue that they were about to occur. And then next we're going to look at the ear. So we're going to look over hearing imbalance, look at the different parts of the ear, and we'll get into what are the organs of hearing, what are the organs or useful parts of the ear for equilibrium and things like that. So make sure to watch that video as well.